Hello children, we'll discuss about reproduction. Reproduction. So first activity is about curdling of milk, and that we need to add a small sample of bacteria from curd into lukewarm milk. Because bacteria needs certain temperature for dividing. It cannot divide extremely at room temperature, also a little more than room temperature is required. That's why we need to add the bacterial sample from curd to lukewarm milk. Okay. Next, sexual reproduction. Asexual reproduction. Asexual reproduction. In asexual reproduction, children, we know that there are different modes of asexual reproduction. In asexual reproduction, there is no gamete formation, no fusion of gametes, except parthenogenesis. What are the different modes? Binary fission. In binary fission, single organism divides into two organisms. In case of multiple fission, single organism will divide into so many organisms. This can be seen in the case of paramecium or mycoplasma. Multiple fission is seen in mycoplasma also, paramecium also. Next, after binary fission, budding. Budding is seen in yeast. Yeast grows a small bud. In that, the nucleus will develop, and finally the complete organism is formed. So budding in yeast, when budding occurs in yeast, organisms will not separate because yeast would like to live in colonies. Whereas budding in hydra, if hydra budding occurs, the bud that is formed slowly develops the tentacles. It will separate from the parent. It will start living its own life. Okay, so that is budding in hydra. Next, after budding, next one, fragmentation. Fragmentation and regeneration are almost similar. In case of fragmentation in Spirogyra, what will happen? A small fragment from this organism will develop. Spirogyra have small chloroplast. In this way, this chloroplast helps it to makes its own food. This small fragment will develop from the parent, and it develops into complete organism. In case of regeneration in planaria, planaria divides into so many pieces. More than 100 pieces it divides. Each one will divide, each one will grow into a complete organism. So what happens, what is the difference between fragmentation? One small fragments form at once in fragmentation. In regeneration, all fragments are formed at once. After the fragments are formed, there is no parent in case of regeneration, but the parent is there like that only in case of fragmentation. Okay, that is the difference between regeneration and fragmentation. Parthenogenesis. Next one, parthenogenesis, parthenovirgin genesis production. In case of wasp bees, in case of Spirogyra, in case of fungus, you can see this wasp, not only wasp bee, honey bee, different types of bees you can see. What will do? They do is they produce female gametes and male gamete. If fusion will take place, female organism will be produced. If fusion does not take, male gamete is turning into male organism completely. Okay, male organism completely. So this is the difference. This is the parthenogenesis. So in this case, gamete formation is there, but there is no fusion of gametes. Parthenocarpy. If you take parthenocarpy, carpel, from carpel, carpy. Carpel is important here. So virgin carpel. Partheno means virgin, carpy means carpel. Stigma, style, ovary is called as carpel. In this, the ovules will be which is future seed, you know that. But if fertilization take place only, ovule will develop into seed. Here there is no fertilization, directly by supply of auxins and gibberellins. Auxin in little amount, gibberellin in very good amount. So major gibberellin, secondary auxin also is required for growth. Okay. So when these are supplied, this fertilization need not occur, and directly this ovary develops into a fruit which does not have seeds. Okay, fruits, seedless fruit will be formed in case of watermelon. In case of papaya, we can see, when I am small, I saw that papaya has so many seeds, hundreds of seeds inside the papaya. But now there are no seeds. If you cut open the papaya, there are very few seeds or no seeds. That is because of this parthenocarpy only. Tissue culture. Tissue culture means take any plant, take any part of plant, cut it into small pieces and put it in the culture medium. This culture medium consists of plant hormones and nutrients. Slowly the embryos will develop inside this. Now you need to sow it in soil, directly into the soil, and the plants will grow. Small seedlings will start growing. So as many plant parts are available, those many plants we can produce. So it is a method by which so many number of plants can be produced in very short time. Next one, spores. Spores are present in fungus, ferns and also moss plants. Spores are present in fungus. Fungus will, suppose if you take the bread mold, this is bread mold, there are root-like structures. So these are mycelium which grows. This is called as sporangium, and sporangium grows into, when sporangium is completely mature, it will burst open and release all the spores outside. Each spore falls on any substratum like food material, it grows into a complete organism. If you take fern, moss plants are small half or 1 centimeter plants that can grow on rocks. They have sporangium which looks like flower. If someone looks from distant, they feel that this is flower because it is colorful, but it is actually sporangium. Inside the spores are there. Whenever it bursts open, all the spores will release out and each spore can grow into a complete plant. This is moss plant. This is a moss plant. In case of fern, fern will have leaves. Each leaf has, if you turn underside, you will see that there are sorus. This sorus has all the sporangium, and sporangium has spores. So spores are present inside the sorus for this fern plants. This type of leaf which is having, all the leaf has spores. So all the leaves of fern plant are called as sporophyll. They bear, the leaves bear spores, that's why they are called as sporophyll. This is about different modes of reproduction, asexual reproduction. Next, vegetative reproduction. What is a vegetative reproduction? Reproduction from, what is reproductive part of plant? A reproductive part of plant. Reproductive part of plant is flower. What are non-reproductive parts? They are vegetative parts. Vegetative parts of plants are stem, leaves, roots. All these. Okay. Stem, leaf, roots are called as non-reproductive parts. Stem, you know that leaves, bryophyllum is a plant that reproduces by leaves. Roots, you know so many examples for plants that reproduce by roots. For example, if you take muraya means karvepaku. This plant, see, usually this is a plant, roots are growing in different direction. Each root has capacity to produce new plant. So that's why near the karvepaku plant, you can see that there are so many small plants growing. Even Goa plant also can grow like this. Other plants like Millingtona also can reproduce. Millingtona means Tigamalli. Tigamalli. This is also growing in the same way. Roots develop small stems and branches will. This stem will grow branches and leaves are formed. In this way, roots can give rise to a complete plant. Stem. There are so many varieties of stems. For example, if you take runner. All grass plants are runners because they grow their stem horizontally, and leaves will grow this vertically like this. Wherever there is leaves formation, their root formation also is there. This type of plants are called runners. And stolon. 1 minute. Okay, this is runner plant. Next one, stolon. What is a stolon? In case of stolon, it's like this. The plant, like strawberry, will grow leaves. After the leaves are formed, then it will bend down. This stem will bend down and roots are formed. Here already leaves are formed. This can grow into a complete plant. That is the difference between the runner and stolon. Next, rhizome. Rhizome, example for rhizome is turmeric, ginger. Plants grow on. This is a stem that is growing underground. These are scaly leaves. And from this there is stem growth, leaves growth, roots are growing. This is rhizome. In case of corm, you take for example chamaganda, colocasia. These are all scales. Here the leaves are growing, here the roots are there, in this way. So this is vertical, this is horizontal, this is vertical. That is the difference between rhizome and corm. After that, bulb. Bulb example, this onion, Allium cepa. A small branch will grow for this, and this will grow the stem, and this can grow into an individual plant. In this way, onion grows. This is bulb. And tuber, for example, if you take the potato roots will be there underground. They are underground stems also. Underground stems store the food material and bulge out, which is called as potato. This potato has eyes over the potato. You can take this potato and sow it in the ground. It can grow into a complete, so many plants. How many eyes are there? Those many plants will be formed from the potato. So that is about it. Different types of vegetative growth from stem region. Next one is artificial vegetative propagation. We can also grow some plants vegetatively. For example, if you take the plants like rose, hibiscus, over the stem there are small buds. These are the buds which have capacity to grow roots. So you cut the plant here and sow it in soil. This part will be below the soil. Here one bud is there. It will gradually develop the roots and it can grow into a complete plant. So what is the condition for that is you need to choose the plant such a way that there are buds available for that, and stem buds. These are stem buds. So they are present in plants like Muraya, Karvepaku or hibiscus, rose, like these plants. Next, if you take the plants like jasmine, you bend one of the stem and put some soil over that. After a few days, this roots are formed and they can grow into an individual plant. This is cutting. This is layering. A third type of vegetative propagation, suppose you take two mango plants in such a way that one is very good in more fruits, it is producing more fruits. Second plant is producing more juicy fruits. So what we'll do is we'll cut not straight but in V shape. Here also we are cutting this plant in V shape, and one part of the plant that is attached to soil, one part of the plant that is above the soil. This is called as scion and this is called as stalk. Both of them will combine, will grow it into a new plant by tying them very hard, supplying with them wherever there it is joined, they don't simply tie the cloth. This is stalk, this is scion. So here when they tie the cloth, they'll take some sawdust, so that that will be helpful to hold some moisture and helping the regrowth of new tissues between the stalk and scion. That is about, okay, we'll see the sexual reproduction in plants. What is the reproductive part of plant? I said you reproductive part of plant is flower. What are the parts of flower? Flower has parts like this. This is flower. Okay, so this is calyx, this is corolla, and anther and filament together form androecium or stamen. It is male reproductive part of flower. Next, stigma, style and ovary. This one forms the female part of the flower. Okay, this stigma, style and ovary forms the female part of the flower. This is stigma, this is style, this is ovary. Inside the ovary there are So many ovules. Okay. This is called as, this female part of flower is called as gynoecium. Gynoecium is also called as carpel. So we saw in Parthenocarpy, right? It is called as Parthenocarpy. I said you it is a virgin carpel. That's why it is called as a Parthenocarpy. So how this sexual reproduction occurs? So for that we need to understand the structure of ovule, stigma, style, ovary. So inside ovary there are so many ovules. As many number of ovules are there, that many pollen grains will fall on the stigma. That many for pollen tubes are formed. More than that, pollen grains will fall on stigma. But that many pollen tubes will be formed from the stigma. So we cannot show those many ovules inside our ovary. So what I am doing is, I will show you only a single ovule. This is the ovule that is present in our seed. And this will form, this is having the internal microsporangia. That is a very important part of the ovule. This microsporangia is covered by layers. These layers that covers the microsporangia are called as integuments. Integuments. And inside this there are three microsporangia there. Oh, okay. Integuments leave a small gap in the between the lower region. This is called as micropyle. Micropyle, through which the pollen tube enters. The cells that are present in microsporangia, ovule, what is it called as? Megaspore. Megaspore. These three cells are called as antipodals, and this side there are two cells called as synergids, and in between there is an egg cell. This is egg cell. And here there is a central cell. Central cell has two nucleus, remaining all are having only one nucleus. This is central cell. Okay, when pollen grain falls here, pollen grain has two nucleus. When pollen grain falls here and it develops a pollen tube, this pollen tube extends till here and it grows till the micropyle. At the micropyle end, the two nucleus will be released inside the megaspore. What happens next? Three antipodals, one egg cell and two synergids. Sorry, the central cell. This is egg cell. Okay, synergids, antipodals. Now, when the two nucleus are released from pollen grain, one will fuse with this egg cell, one will fuse with this central cell. This will develop to form zygote and this will develop to form endosperm. Endosperm is a future cotyledons. Cotyledons are present in plants. Zygote develops to form embryo. Where is this embryo and cotyledon in the seed? If you take the, for example, bean seed, here there is a small sprout that sprouts that come out will initially present in the form of small structure, and this is cotyledon. This is cotyledon which nourishes this small embryo. Before the seed will start forming small leaves. After leaves are forming, it makes its own food. Before that, plant requires nutrients to grow this embryo. So this small embryo, which is later sprouts out and forms sprouts, from that seedlings will be formed. Okay, this is embryo. This embryo will have plumule, the upper portion, and radicle, the lower portion. Radicle is developing to form root and plumule is developing to form shoot. Okay, that is about reproduction in plants. Okay, since two fertilizations are taking place here, this is called as double fertilization. So the remaining parts, the remaining topics of the lesson are male reproductive system, female reproductive system, and cell division. This I'll say you in the next video. Okay children, next topic is Reproductive system, male reproductive system. Male reproductive system, the major organ is testis. Inside this testis there are small lobules. Inside these lobules there are seminiferous tubules. In these seminiferous tubules the sperms are produced and they are collected by one small duct here. This duct is called as vas efferentia. Vas efferentia. The seminiferous tubules will produce the sperm. They are collected by vas efferentia. From vas efferentia they pass to a small storage sac here called as epididymis. Temporarily sperm is stored inside the epididymis and then it passes to vas deferens. Vas deferens. From vas deferens it is going forward. So the structure of male reproductive system is like this. Scrotal sac is present. The scrotal sac is the place where both testes are present. Why? This helps in lowering the body temperature. Lowering body temperature by 2 to 3 degrees Celsius, which helps in maintaining a little lower temperature for the survival of sperm. Sperm requires a little lower temperature for the survival. Okay. Next thing. Here one testis is there. Here one testis is there. This is epididymis. Epididymis. And from here extends the vas deferens. Vas deferens extends. And here we have urinary bladder. And here we have connected seminal vesicles. So this duct will pass over this duct, which is urethra. First of all, here urethra is present. So this duct will pass over the urethra. The vas deferens will pass over urethra and then it will connect to the duct that is coming from seminal vesicle. So this is also passing over urethra and here also seminal vesicles are present, that is seminal duct. So this will connect like this and a small duct will be formed here. This is the duct that connects seminal vesicles and this all is called as ejaculatory duct. After that here the penis and all is present. Inside this penis the urethra, which is long in males, is present. This is urethra is also called as urinogenital duct. Urinogenital duct. Urinogenital duct is a common passage. Urinary bladder, urethra is present. So this is the passage for urine also. At the same time the liquid that is coming from all these parts. This is seminal vesicle will secrete seminal fluid. Here prostate glands are present. Prostate fluid is secreted. Here small corpus glands are present. Corpus fluid is secreted. What is the importance of seminal vesicles? When the sperm goes out of the body, it needs to survive for few days. So for that, energy required is supplied by this seminal fluid. When the sperm is present inside the body, for the sperm energy is required. That is provided by prostate gland. Corpus glands. When urine is passing through this, because of capillary action, some of the urine may remain at the walls of this urethra. So there may be acidity. Sperm is a very sensitive cell. It dies very fast. So to avoid that, the corpus gland neutralizes the acid that is present in the walls of urethra and helps in the movement of sperm. So now the semen. What is, first of all, what is seminal fluid plus prostate fluid plus corpus fluid forms seminal plasma. Seminal plasma. This seminal plasma plus sperm forms the semen. Okay. So the fluid from this and this three glands will form seminal plasma. Seminal plasma plus sperm that is passing through this, together it forms the semen. Okay. What is the structure of sperm? Sperm structure is a very important question, children. Structure of sperm. Sperm has a head region, a neck region, a middle piece, and a tail. Tail helps it to swim. Middle piece has so many mitochondria. These are all mitochondria that supplies with it with the energy. And in the head region there is nucleus and the acrosome that is required for sperm to penetrate into ovum is also present here. Okay, this is about the structure of sperm. 40% of sperm has high motility, whereas 60% of sperm is very slow. The production of sperm starts at adolescence age and it continues almost, the rate may reduce, amount of sperm that is produced will reduce, but it continues as long as the male organism is existing. So that is about it. Female reproductive system. Female reproductive system. So this is a rough structure of female reproductive system. Female reproductive system consists of, these are ovaries, these are fallopian tubules, and this is uterus. After uterus, this is cervix. Oh, sorry. Here cervix is there. This is cervix and this is vagina. Cervix and this is vagina. Okay. These are the parts of female reproductive system. When sperm enters into female body, this is the way, and ovum will enter into fallopian tubule by this way. So this is the sperm will pass through this direction, ovum will pass through this direction, and here the fertilization takes place. Where the fertilization takes place in female body, in the fallopian tubule. This is the region where the fertilization of organisms, fertilization of the gametes takes place. How the ovum is released. Ovum release also has a procedure, children. Inside the ovary, first of all, the smallest cell that is called as follicle is formed. This is a primary follicle. When it matures, inside this ovum will be formed, and this ovum releases out of the ovary. This is called as ovulation. Ovulation occurs on the 14th day of menstrual cycle, approximately on the 14th day of the menstrual cycle, and after that this Graafian follicle will be slowly it will disappear, and it is called as corpus luteum. Corpus luteum. Okay. This is primary follicle and this is Graafian follicle. This is ovulation. After ovulation, the ovum enters into the fallopian tubule and fertilization occurs inside the fallopian tubule. There are two things that can happen in the female reproductive system. One, fertilization occurs. If fertilization occurs, this is a ovary. I'm drawing only ovary, children. If fertilization occurs, the ovum will pass, the zygote will pass into the uterus and it will fuse, that zygote will fuse into the inner wall of uterus called as endometrium. Endometrium thickens itself during the first 14 days of menstrual cycle so that it is ready to receive the zygote. Okay. This zygote that is formed inside the fallopian tubule enters into the uterus and it fuses with the uterine walls, in endometrium of uterine walls, and this is called as implantation. Implantation occurs, then it leads to pregnancy and all. Second thing that can happen if fertilization did not occur, the endometrium wall that is thickened will wait for the implantation process. If the implantation did not occur after more days, 14 plus 14, 14th day ovulation occurred. After that 14 days the uterus will wait for the ovulation, sorry, implantation to occur. Okay, first 14 days is ovulation, I said you. First 14 days, after first 14 days ovulation occurs. After the 40 ovulation is completed, this uterus will wait for 14 days. So totally it is 28-day menstrual cycle. After the 28 days, this endometrium will shed out through the cervix, through vagina, and it is flowing out of the body. This is called as menstruation. So there are two fates. Either fertilization occurs or fertilization does not occur. What happens if implantation occurs? If implantation occurs, there are four membranes that are formed in the female body. What are the four membranes that are formed in the female body? First, this is uterine walls, endometrium. Uh, the zygote that is formed will fuse with the endometrium. It enters inside. When it enters inside, the finger like projections are formed around this newly formed zygote, and this layer is called as chorion layer. The chorion layer along with the uterine cells will fuse together. The fusion of this will take place and it will develop to form placenta. Placenta is supplied with so many blood vessels. There is a layer, placental cells are formed by combination of chorion that is formed from uh, uh, the newly formed embryo and the mother's uterine cells. So there is a layer that separates the parental cells and as well as the daughter cells. Daughter cells and parent cells are separated from each other by a small layer that is present between placenta as well as the baby. Okay, so there is a small membrane that separates this placenta from the baby, but this is the placenta is the very important organ that is present inside the what is it, a female reproductive system, because it nourishes the baby. The growing baby gets nourished. It is formed after 12 weeks after the pregnancy, and this is very important for nourishing the baby. It is supplied with rich amount of blood vessels, and it acts as a medium for supplying all the nutrients between mother and child. And waste materials also pass from baby to mother through this placenta in the initial stages, and later on this baby that is developed slowly forms a small layer around itself, and the baby starts developing here. This is baby that is developing, and slowly one layer is formed. This is the third layer that is called as amnion. Around this amnion, around this baby, amnion is formed. Inside this there is a fluid called as amniotic fluid. Okay, this fluid protects the baby from any shocks and injuries, and during the development of amnion only, from this placenta one small structure will develop that is called as allantois. Allantois is a structure that develops to form umbilical cord. Umbilical cord is a cord that helps in connecting baby with the mother, and all the nutrients that are initially passing through placenta to baby are now passing through this umbilical cord. Umbilical cord acts as a medium of transport. It is a cord of transport between mother and baby. One more structure will be formed, yolk sac. This yolk sac has no use for human beings. Actually, yolk sac is a structure that is present in oviparous organisms. In oviparous organisms, it acts uh, as a part of nourishment. For us, we have placenta and nourishment is supplied through placenta, so there is no use of yolk sac. This is one of the vestigial organ that is present in our body. Okay, so this is about the developmental layers. Four layers: chorion, placenta, allantois, and amnion. Four layers are formed. Four membranes are formed during developmental stages. After that, what is present after that? After the formation of the complete baby, the uterine walls, the baby is upside down during the development, complete development. When the development is completely completed, the baby will turn upside down, head is down and legs are down. This is hands. So this will be the baby will be here like this. Legs will be present here and hands will be present here. This is the head region. So baby will turn upside down and ready for the pregnancy. At that time, uterine walls will start producing contractions and relaxations. This will lead to pain called as labor pains, and labor pains lead to childbirth. Baby passes slowly through the cervix. Cervix by that time it becomes very wide, and baby easily passes through the cervix, and after that through vagina, and it comes out. When baby comes out, along with the baby is attached to a cord called umbilical cord, and placenta is also present. So this placenta and umbilical cord also come out of the mother's womb, and these are called as afterbirth. They need to be cut and separated, and a clip is attached to the baby's belly, so that this part is separated and a small part attaches to the baby's belly. That dries out very soon. Second thing is colostrum starts developing in mother's mammary glands. This is a yellow color fluid, pale yellow color fluid, not thick yellow, pale yellow color fluid, which is rich in antibodies. These antibodies are very good for providing immunity to the baby. Till now baby is not contact with the external world. Mother's womb is protecting her from any kind of infectious agents that are present in the external environment. So this colostrum is very important to develop immunity in the baby. So the first 1 hour of baby's birth, colostrum is definitely felt. Okay, the first milk that is formed in the mother's mammary glands is called as colostrum. It is seen in animals also. It is called as junnupalu. So many of us eat it as a sweet. So this is a very good what do you say, nutritious food. Next, cell division. Cell division is also a very important topic. How does cell division occurs? Cell division is two methods: mitosis and meiosis. Mitosis and meiosis. Meiosis is very less discussed in your textbooks. Mitosis is very deeply expressed. I mean, it is given detailed. Mitotic cell cycle is detailed. What is present in mitotic cell cycle? The events of mitotic cells are divided into G1 phase, S phase, G2 phase, M phase. There are 4 phases. What happens in G1 phase? Gap 1 phase. Gap 1 phase. Okay. G2 is gap 2 phase. In between S phase is synthesis. Synthesis of DNA. Equal amount of DNA. How much DNA is present? We have 23 pairs of chromosomes. Again 23 pairs of chromosomes are produced during this S phase. And this is the gap phase between this one cell cycle is completed. After that there is a small gap. Okay, that gap phase, cell prepares itself for the production of DNA. Okay, that phase is called as G1 phase. After the DNA is produces, again cell takes some gap to produce to make itself ready for mitosis, mitotic cycle. The mitotic cycle is the actual phase where the cell division occurs. It is divided into two steps: the karyokinesis. Karyokinesis means nuclear division. Nucleus divides. After that, cytokinesis. Cytokinesis means cytoplasm divides. What happens in karyokinesis? Karyokinesis is divided into 4 phases: prophase, metaphase, anaphase, telophase. PMAT. What happens during these 4 phases? Very important. During prophase, cell makes itself ready for cell cycle. The nucleus, all the organelles that are present inside the cell will disappear. Nucleus, around the nucleus there is nuclear membrane, that will disappear. Inside nucleolus is present, that will disappear. And the chromatin material, which actually is condensed, it will turn into chromosomes. Okay, so these are the main events that are taking place in prophase. After that, metaphase. All these chromosomes that are formed will assemble themselves in equatorial plate. Equatorial plate means central, like Earth has an equator that is going through the center of the Earth. Same like that, the cell also has an equatorial plane where all the chromosomes will assemble. Thus, that happens in metaphase. In anaphase, these chromosomes which are assembled will split into two parts. All of them will, oh sorry, yes. Second thing that happens in mitosis, there are spindle fiber formation. All the spindle fibers will form. Spindle fibers are formed from centrioles. Centrioles will develop small fiber-like structures and they extend till centromere of chromosomes. The central part of chromosome are called as centromeres. Okay. In anaphase, the splitting of chromosomes take place. Chromosomes split and they start moving to the opposite ends. In last phase, that is telophase, all the changes that took place in prophase will be reversed. That is, all chromosomes that move to corner will condense again, nuclear membrane will form, nucleolus will form, and two nucleus are there, so two nucleolus will form, and slowly the centrioles which are present, one centriole is here, one centriole is here, these centriole will start moving to the center of cell and all the organelles will reform again. These changes are reverted in the last phase, that is telophase. With this telophase, the nucleus are divided. After that, the cytoplasm will start a small constriction and gradually the cytoplasm will divide in such a way that organelles are divided equally between the cells. Okay, this is cell cycle. After cell cycle, meiosis. What happens in meiosis? All this process. In case of meiosis, what will happen? G1 phase, S phase, G2 phase will take place. But meiotic phase 1 occurs, meiotic phase 2 occurs. At the end of meiotic phase 1, two daughter cells are formed. At the end of meiotic phase 2, these two will again divide and four daughter cells are formed. What is the problem with this is, it's not problem actually, it is the advantage with this is the synthesis of DNA take place only once. That means 23 pairs of chromosomes are present in our body. Again 23 pairs are formed. But division is occurring 4 times. So what will happen after, at the end of meiosis 1, 23 pairs will enter here, 23 pairs will enter here. At the end of meiosis 2, only 23, 23, 23, 23. That means here paired structures. That means 2n stage. 2n means all are in pairs. In case of this, they are not paired. Unpaired, each one is unpaired. Each cell is unpaired, is having unpaired chromosome. So the paired chromosome state is called as diploid stage. Two sets of chromosomes are present. Unpaired state is called as haploid state. Okay. So that is the difference between meiosis and mitosis. At the end of meiosis, four daughter cells are formed. Each one is having only one set of chromosomes. The doubling of chromosomes take place only once. So you need to remember only four differences, children. They will not ask you this for an 8-mark question. Maximum they'll ask you for a 2-mark question. If they ask for 2-mark question, you simply say that meiosis takes place for the formation of gametes. Mitosis takes place in the entire rest of the body. Okay. And pregnancy. Contraceptive is anything that is used to prevent pregnancy. So contraceptives are present in the form of physical devices or in the form of pills. Pills means tablets that can be taken every day, that can be taken monthly once. Different types of pills are available. Apart from that, surgical methods are also present. Vasectomy. Vasectomy means vas deferens. From testis, vas deferens extends, which carries the sperm. So to prevent pregnancy, simply this vas deferens is cut and it is tied. Okay. In this process, sperm cannot pass out of the body. Since sperm will not pass out of the body, it will not be reaching the female body. In case of tubectomy occurs in female body. The fallopian tubule, that is the place where the fertilization occurs, is cut and here it is ligated. So there is no chance for entry of ovum into this. Even if it enters, it stops here itself. It can't go till the place where sperm reaches and fertilization occurs. The sperm cannot pass through this it cannot reach the ovum, and fertilization will be prevented, and this can be useful to prevent the pregnancy. So that is about this reproduction chapter, children. If you still have any doubts, you can ask me through my WhatsApp number.